Hi, I am Aurel Enriquez, and this presentation contains our discussion on the week 11 topics, which would be our introduction to virology. So ayan po, to my students, welcome to virology. So dito po natin sisimulan, no? Tapos na tayo sa mycology. We're done with the study of clinically significant fungi. So let's now talk about clinical virology for our finals period. Now, before we actually get into the discussion of the concepts or topics that we need to understand for our introduction to virology, let's first have this point of view. Let's first understand what is the importance of studying clinical virology, bakit ba natin kailangang aralin ito? And let's look at that by going through these different viral pandemics that happened in the past 40 years. Okay, so ano yung iba't ibang mga diseases that were caused by different viruses, that became a public health concern in the past 40 years. First off, in 1981, there was a global spread of HIV, or the human immunodeficiency virus. Now, during that time, it was recorded that there was approximately 79 million people that were infected, and this led to 36 million deaths. But of course, eventually, antiretroviral drugs were formulated, and antiretroviral therapy, lead to the decreased mortality rates. So when we get to our week 16 discussion, we'll talk more about how does the human immunodeficiency virus cause what it does to the people. How does it lead to mortality? Next, in 1999, there was also a spread of the West Nile virus. Now, the West Nile virus, this is an arthropod-borne infection. All right, so merong insect or mosquito vector ang West Nile virus that led to its spread to other people. And as we can see right here, there has been a resurgence in 2012. So medyo umangat ulit yung cases pagdating ng 2012. After that, the dengue fever virus also became a public health concern. And then in 2002, we have the SARS-CoV-1, all right? The severe acute respiratory syndrome coronavirus 1. So, meron pong version 1. Now, meron pong version 1 ang SARS-CoV, all right? So, that happened in 2002, but it wasn't as serious as what we experienced with SARS-CoV-2 and COVID-19, because the spread of the virus was immediately halted. Maagang napigilan yung pag-spread ng virus to more countries, because the countries back then, even here in the Philippines, they were very quick with closing the borders, so infected people weren't able to further travel to other places to spread the infection. So ayun nga po, SARS-CoV-1 yung kumbaga parang first version ng SARS-CoV-2, which again, that happened in 2002 hanggang 2003. Next, in 2007, there was also the emergence of the Zika virus, right? So the Zika virus is also one of those arboviruses or arthropod-borne viruses. Now, the problem with Zika virus is this has been linked to microencephaly in neonates or newborns. Microencephaly meaning they have a smaller size of their head, compared to normal newborns. We'll talk more about that also when we get to week 16. Next, in 2009, there was also the spread of the influenza H1N1 virus. We'll also talk more about this when we get to our conversations or discussion about the influenza viruses. But wait, there's more. Okay, so madami pa po. Next, in 2012, there has been an emergence of the MERS-CoV virus, or the Middle East Respiratory Syndrome coronavirus. So this one is a more zoonotic type of virus. Now, it was able to spread to 27 different countries. 2,000 cases were noted, and 866 deaths were recorded. All right, so this is also something that we'll talk more about when we get to our discussion on coronavirus. Next is chikungunya, which became a public health concern in 2013. And then in 2014, there was an outbreak of Ebola, not the Ebola virus. So this happened in 10 different countries and around 28,000 cases. So that's a lot. That's a lot, considering that Ebola virus is not a very common type of um, disease or virus or infection that usually just spreads out everywhere. Next, in 2019, we all know this. No, we all know this. In 2019, the SARS-CoV-2 became a again, ayun nga, no, it became a public health concern and eventually caused a global pandemic. So as of now, no, there has been a total of 6,332,963 deaths and still counting, guys. The pandemic is still going on. All right, we just kind of like lifted the alert levels, no, nagluwag lang ng mga restrictions and other stuff like that, because it's not as kind of like hindi na ganun karami yung people who are being reported to be infected with the virus compared to what happened in 2020. But the pandemic or the spread of this virus is still very much out there. It's still ongoing. So as you can see right here, the virus is still mutating rapidly. So there could be different strains out there that could, again, spread to more people, cause more serious infections, right? So next, the monkeypox virus also took the spotlight, but the monkeypox virus was not that kind of like a serious concern, no. Usually if a person has been vaccinated against smallpox vaccine prevented the monkeypox infection, or at least the infection was not as severe for people who had the smallpox vaccine. All right, so let's now get into the main topics of our week 11 discussion. So let's first go through the general characteristics of viruses, specifically involving their structure and their taxonomic classification. Then after that, let's talk about viral replication. So how do these viruses reproduce? Let's proceed with the general characteristics. Now, one of the most basic questions that has been raised by people who are new to the field of virology is that, are viruses living things or are viruses non-living things? All right? So ano ba talaga, no, ang mga virus, sila ba ay living organisms or non-living organisms? And I think the answer to that would lie in our understanding of their life cycle, all right? How they replicate. Now, viruses, if we were to use oversimplification, no, we oversimplify po natin, viruses are literally just a shell that carries genetic material or their instructions for replication. So on their own, they don't have any organelles that can perform any processes that is necessary for their survival. All right, so on their own, they can't acquire nutrients, they can't produce waste materials, they can't multiply by themselves. So they are known as obligate intracellular parasites. They require to be inside of the host cells before they're capable of doing anything. So viruses, they can't do anything by themselves. So viruses can't be left out there and expect them to survive and behave like free living organisms, diba? Hindi katulad ng mga ibang organisms like parasites or bacteria. There are parasites and bacteria out there that are called free living, diba? So even if they don't find a human host, it's okay because they could thrive and grow in the environment. So it's not like that for viruses. Now, there's only a certain amount of time where they would still be viable before they have to find their preferred host and ultimately get into their target cell and infect that host. So, ayun po, no? Kumbaga, is parang requirement sa kanila, no? Na makahanap ng host, or else, no? They would cease to survive. They would cease to exist, all right? So, clinically significant man for humans or hindi, all viruses are obligate intracellular parasites. Lahat po ng viruses are obligate intracellular organisms. So, they may still be viable outside of their preferred host for a few hours or maybe a couple of days at max, but it is guaranteed that they will not be able to perform any function that is necessary or that is inherent, no, to other living organisms as long as they have not yet entered their respective host. Ulitin ko po, wala silang kayang gawin mag-isa unless naabot na nila, no, or nakapasok na sila sa target host cell nila. Alright, so by themselves, viruses, by themselves, no, we can't say that they're actual living organisms. But once they enter their appropriate host cells, then yes, okay? Only then would they start behaving like an actual living organism. Also, they can't multiply by binary fission. All right, they can't do that. So what they do is that they enter a living host cell and then they control or manipulate the host cell function to replicate. Okay, so kumbaga parang minamanipulate or kinokontrol po ng mga viruses yung target cell nila para makapaggawa sila ng copies nila ng sarili nila. So that process is referred to as replication. Okay, so again, viruses do not multiply through binary fission. Di nila kaya yung ginagawa ng mga fungi na nagba-binary fission or nagbabudding, no? Viruses, they only replicate once they enter their preferred host cell, which again proves the point or brings us back to the idea that they are obligate intracellular organisms. Let's talk about the structure of viruses. First is their viral genome. Okay, so pag-usapan po natin yung genetic material that could be found in each virus particle. All right, so the viral genome could either be RNA or DNA, but not both. Okay, ulitin po natin yan, ang mga viruses, no, within them, they could have either strands of DNA or RNA, but it's not possible that they have both DNA and RNA. Maaari po na meron silang dinadala na enzyme that could convert their RNA strands to become DNA, but it's not a usual thing, or it's not a normally observed thing that a virus particle contains both already made RNA and DNA. So ulitin po natin, the viral genome or the genetic material of viruses within their structure is only RNA or DNA, but not both. Okay, so these genetic strands or viral genomes, no, they could either be referred to as single-stranded or double-stranded. Now, what does that mean? People might be confused, no? Hindi ba basta pag sinabi nating RNA, single strand agad? Di ba pag sinabi natin na DNA, double agad dapat yung strands? Let's oversimplify this explanation, no? Kapag sinabi natin na single-stranded, regardless kung DNA or RNA ito, kapag sinabi natin na single-stranded, may literal na may isang strand ng DNA or RNA sa loob ng virus na ito. Okay? So, imagine a virus particle. Pag sinabing double-stranded RNA, ang ibig sabihin, literal, may dalawang strands ng RNA sa loob. Pag sinabing single-stranded DNA, no, it's still a DNA molecule with this, with their double helix structure. It's just that isang kompletong strand lang ng DNA yung meron. Kapag sinabi mong single-stranded. Pero kapag sinabi natin, halimbawa, baliktad, single-stranded RNA, then literal, isang strand lang talaga ng RNA yung meron. Kapag sinabi mo naman na double-stranded DNA, no, literal, meron kang dalawang double helix doon sa looban ng virus. Alright? So I hope nobody's confused about being single-stranded or double-stranded kasi po, di ba, initially, no, when we were taught about the structure of RNA and DNA, alam natin, no, we know that RNA is composed of one strand, DNA is composed of double strands in a double helix formation. But again, no, we have to kind of like learn something new when we talk about viruses. No, maaari nating sabihin double-stranded RNA. So, meaning literal, may dalawang strand ng RNA sa looban. Pwede nating sabihin na single-stranded DNA. So, literal, may isang strand lang ng DNA inside of the virus particle. Alright, so I hope you understood that. Next component of a virus particle is a capsid. So the capsid is a protein coat that encloses the viral genome. So yun nga po sinasabi ko kanina, no? Viruses are nothing more than a container, nothing more than a shell that has either DNA or RNA inside of them. And that's it. So ayun nga po, no? We have what is known as the capsid. So the capsid is the protein coat that encloses the viral genome. So either yung RNA niya or yung DNA niya dito nakalagay sa looban ng capsid. Now, the capsid, this is composed of repeating structural subunits, which is referred to as the capsomeres. Okay, so ulitin po natin yung explanation na yun. Ang capsid, no? Nasa looban nito yung viral genome. Ang capsid ay gawa sa capsomeres. Okay, so ang mga pinagsama-samang capsomeres that would create the capsid. And then inside of the capsid is the viral genome. Alright, now for some viruses, that's all that they are. Okay, wala na silang ibang component. So they're only composed of a viral genome and a capsid. Now that combination together is referred to as a nucleocapsid. Alright? But for some viruses, they have what is known as an envelope. So may dalawang terms tayong pwedeng gamitin, no? A virus could either be enveloped or naked. So kapag sinabi nating naked, yun lang yung component niya. Viral genome, tsaka capsid. Pero kapag sinabi nating enveloped, ano ba ang envelope? The envelope is a phospholipid labile structure that surrounds the virion. Alright? So, yung nucleocapsid na sinasabi natin kanina, no, yung pinagsamang genome tsaka capsid, no, genome sa loob, capsid sa labas, may isang structure pa sa labas nun, which is kind of like an outer layer made up of phospholipids. And that outer structure would now be referred to as an envelope. Okay? Now, another term that we need to be familiar with is a virion. So, kapag sinabi natin virion, this is an entire or complete virus particle. So, kung halimbawa, yung virus ay naked, so it's only composed of the viral genome and the capsid, and that's it, kompletong structure na yun, we could say that that thing is a virion. Pero kapag halimbawa, envelope naman yung virus, genome sa loob, capsid sa labas, tapos envelope sa paligid, no? Tapos yun yung kompletong structure niya. That's also referred to as a virion. Okay? So this this illustration right here, no, gives us kind of like a general structure of what a naked virus versus an envelope virus would look like. So again, nucleic acid sa loob, capsid sa labas. And then that's it for naked viruses. But for envelope viruses, we have what is known as the nucleocapsid. So yung pinagsamang nucleic acid, again, which is either DNA or RNA, nasa looban, and then capsid sa labas. And then after that, may outer structure pa, which is referred to as the envelope. Okay, so I hope you understood all of those terminologies that we just explained. Next, viruses are also classified based on the morphology of their genetic material. So in what manner or in what formation are these genetic materials arranged? Okay, so we have these three different terms. First are helical viruses. So when you say helical viruses, their genome is arranged in a coiled or spiral-like formation, as we can see right here in this illustration. Alright, next, icosahedral or polyhedral viruses, no? Their genome is arranged in a geometric pattern. Okay, a geometric pattern that is made up of about 20, no, approximately 20 triangular sides, as we can see right here in this illustration. Okay, so ulitin natin, kapag sinabing helical parang coil or spiral yung formation ng genome niya. Pero kapag sinabi nating icosahedral or polyhedral, no, it has a kind of like geometric structure made up of multiple triangles. Alright? Next, kapag sinabi naman pong complex yung structure niya, there is no specific pattern or arrangement. So again, we would refer to that as a complex virus. So isa-isahin po natin yung examples na pinakita dito sa figure 64-1. So on the top left, that is a naked icosahedral virus. Notice that very apparent yung geometric shape niya. Now the top right would show us a naked helical virus. Next, on the bottom left, no, this one is an envelope icosahedral virus. So notice yung geometric structure na nakita natin kanina, ngayon nasa loob na siya nung envelope. Okay? Next, same thing, no? If we have a helical envelope virus, just take the helical structure of the genome, put it inside, and then there's an envelope on the outside. Aside from that, no, aside from the lipid envelope, we're also seeing what are referred to as glycoprotein spikes. So in our future discussions, pag-uusapan din natin, no, ano ba ang pakinabang ng mga glycoprotein spikes na to. But for now, just remember, glycoprotein spikes are found on the surface of envelope viruses. Okay, ulitin natin, glycoprotein spikes are found on the surface of enveloped viruses. Okay, so they usually help these envelope viruses to successfully attach to their preferred host cells. Okay. This slide summarizes to us how viruses are taxonomically classified through their order, family, and then their genus and their species. And then listed here are some of the other criteria that we've already discussed, di ba po? So wag niyo pong kakalimutan, no, yung genome type nila, are they RNA or DNA? Ulitin natin, they can't have both. Next, sila ba ay double-stranded or single-stranded? Ilang strands ba nitong mga genetic material ang meron sila? Next, we also have to take note of their capsid morphology. Are they helical, icosahedral, or complex? And last thing that we need to remember is the presence or absence of an envelope. So we also have to keep in mind, itong magkakaibang viruses ba na ito, are they naked or do they have an envelope? Okay, so ayan po. Again, this is the taxonomic classification, or kind of like a simplified version as to how viruses are classified. So yung order po, no, usually the orders, they are made up of different types of families. Alright, so under ng family, no, yung mga family, sa dulo ng name nila, they have the suffix viridae. Okay, so let's take for example Coronaviridae. So sa loob ng Coronaviridae, there are different genera, no, there are different genera of coronaviruses. So sa genera po, no, nakaduktong yung suffix na virus. So coronavirus. So corona, no space, tapos virus. That refers to a genera. And then yung species, these are their common names. So SARS-CoV-1, MERS-CoV, SARS-CoV-2. Yun na po yung mga examples ng species. Alright? So that's how, or this is kind of like a simplified version on how viruses are taxonomically classified. Let's proceed with viral replication. Now, as I've mentioned earlier, viruses are obligate intracellular organisms, and it is a requirement for them to first be inside of their target host cell before they're capable of replication. So let's now discuss the steps in the viral replication cycle, or otherwise referred to as the infectious cycle. So dito po matututunan natin, no, paano nagmumultiply yung mga viruses. Kanina, we learned about their structure. So we learned that they're only composed of a capsid and a genome. And then for other viruses, there's this addition of an envelope. So with just that, how are they capable of multiplying? So viruses, again, they are strict intracellular organisms that rely on the components of the host cell to replicate, and therefore they're only capable of replication when they're inside of the host cell. So there are 6 steps to viral replication, and again, this is otherwise referred to as the infectious cycle. So the first step is attachment or adhesion or absorption. So all of these 3 terms may be used interchangeably. All right. So again, we have the term absorption, adhesion, and attachment, which again is the first step in the infectious cycle. So absorption involves recognition of a suitable host cell and a specific binding between the viral capsid proteins or glycoprotein spikes. So kung halimbawa naked po yung virus, no, the capsid proteins needs to bind with the receptor on the surface of the host cell. Now kung envelope naman po yung virus, then yung glycoprotein spike sa ibabaw yung magbabind sa receptor doon sa preferred host cell niya. Okay? So ayun po. Next, no, again, to simplify, it's the glycoprotein spikes or yung capsid proteins kung naked yung virus, no, plus a matching carbohydrate receptor on the surface of the host cell would lead to successful attachment. All right. Next, the second step to that would be, the next step to that would be penetration. So on the next slide, meron po tayong illustration na makikita to guide us along these steps. But for now, i-define muna po natin, no, yung bawat step. So for naked viruses, the way that they enter the host cell, no, or yung method of penetration nila is via direct injection. So they just directly enter the host cell. But if the virus is enveloped, then it could either fuse its envelope with the host cell membrane, or it would be engulfed or somewhat phagocytized by the host cell. Alright? So, ulitin natin yung dalawang naunang steps. First is absorption. So, sa absorption, magbabind either yung glycoprotein spike or viral capsid nung virus, and then magbabind siya dun sa receptor dun sa host cell. After that, yung penetration, kung naked yung virus, diretso na lang siyang makakapasok inside. But if, for example, the virus is enveloped, no, either yung envelope niya ay mag-fuse dun sa membrane ng host cell or i-phagocytize siya ng host cell pagkatapos mag-bind ng receptor and glycoprotein spike nila. Alright, next is step 3, which is uncoating. So uncoating, this is the process by which the capsid is removed. Okay? So ayan po, no, ulitin natin, pagdating sa looban ng host cell, ang mahalaga dito, ang mahalaga po ay magamit yung viral genome sa looban ng virus or viral capsid. So you have to destroy the capsid. So the capsid is removed either by degradation of viral enzymes or host enzymes. Alright? So ulitin natin, either may enzymes dun sa looban ng host cell na kayang i-dissolve yung capsid, or may sariling enzyme yung virus para pagpasok niya dun sa cell, i-dissolve niya yung sarili niya capsid. Alright? Or sometimes it could also happen by simple dissociation. So kumbaga parang automatic na nakaprogram na pagpasok sa loob ng host cell, maghihiwa-hiwalay or masisira yung structural integrity ng capsid. Alright, so that's what happens in uncoating. So uncoating is necessary to release the viral genome for the delivery of the viral DNA or RNA to its intracellular site of replication. So yung DNA or RNA po ng viruses, no, they could either be used, no, maaaring gamitin sila doon sa nucleus ng host cell, or maaaring naman doon sila gagamitin sa cytoplasm ng host cell. So iba-iba rin po yun per virus. But again, the third step, no, in the infectious cycle is that you remove the capsid through uncoating para mapakinabangan yung genetic material on the inside. Okay, next po, no, the fourth step is the eclipse stage or the macromolecular synthesis stage. Again, fourth step in the infectious cycle is the eclipse stage or the phase of macromolecular synthesis. Now, during this stage, the virus will transcribe and synthesize its mRNA or messenger RNA, which would now encode the proteins that are necessary to create new virus particles. So, sa eclipse stage po, no, dito yung part kung saan gagamitin na nung virus yung dala-dala niya na genome and use it as kind of like an instructional manual for the host cell. So, it's literally going to tell the host cell to stop whatever they're doing and start doing kung ano man yung sinasabi dun sa genetic material ng virus. Now, it's going to start giving orders to make these specific proteins to create more copies of the virus. Okay, so dito po sa process na to, yung virus na yung nasusunod kung ano yung gagawin ng host cells. After that, fifth stage is assembly or the maturation stage. So after the host cell successfully follows the viral genome's instructions and after nilang masynthesize lahat ng proteins that is needed to create new viral particles, then just literally, these proteins would assemble and pack into new nucleocapsids. All right. So the sixth and final step is called the release. Alright? So, katulad din po ng penetration, dito sa release, magkakaiba din yung methods whether the virus is naked or enveloped. So, for naked viruses, they could either go through direct release. So, exit na lang sila basta-basta dun sa host cell and then they'll move on to infect the neighboring cells or yung mga katabi na healthy pa na cells. Alright? Another option for that could be naked viruses could also go through apoptosis or cell lysis. So literally, they'll destroy the host cells. Sisirain nila yung host cell to let themselves out. All right. Now, if the virus is enveloped, ano pong nabanggit natin sa discussion kanina when we were talking about general structure of enveloped viruses? Enveloped viruses, their envelopes are they kind of have like this phospholipid structure that is usually sourced from the host. Okay? Ang envelopes po ng mga envelope viruses ay galing din dun sa host cell nila. And for them to be able to take a part of the host cell membrane, they would go through the process of budding. Alright? So on their way out, they're going to take a piece of the host cell membrane, take that, and that would be their new envelope. Okay, so basically, inutusan mo na nga yung host cell na gumawa ng proteins para sa'yo, i-assemble to para sa'yo. Tapos on your way out, kukunin mo pa yung piraso nung host cell to become your envelope. Alright, so that's what happens in budding. So ayun po, sana wag malito, magkaiba yung meaning ng word na budding when we're talking about mycology versus when we're talking about virology. So in mycology, budding refers to the replication or binary fission of yeast cells or fungal cells. But in the viral replication cycle, when we say budding, this is the method of release or exiting for enveloped viruses. This line gives us a visual on how each of the steps in the infectious cycle would happen. All right, so first up is attachment. So this is where either the glycoprotein spikes or the viral capsid proteins would recognize a carbohydrate receptor on the surface of the host cell so they could attach or bind to one another. The next step to that would be penetration. So either it's a direct injection for naked viruses or through fusion if the virus is enveloped. Next is uncoating. So in uncoating, the viral genome is now released onto the cell cytoplasm by dissolving the capsid. Alright, so alalahanin natin yung basic structure kanina. Genome sa loob, capsid on the outside, and then excess layer lang kung meron pang envelope. So for those types of viruses, after nung penetration step, next is uncoating. So what would happen is made-dissolve yung capsid para ma-expose yung viral genome. Next is the eclipse stage, or again, the stage of macromolecular synthesis. So this is when the host cells own functions would stop and they would begin to take instructions from the viral genome. So this would lead to the production of proteins that are necessary to make new virus particles. So sa macromolecular synthesis po, kung ano man yung original function nitong host cell na to, ititigil na niya yun. Kung ano man yung inuutos ng viral genome, ng virus na dumating, yun na yung susundin niya. So, ang mangyayari, it now just creates proteins to make more virus particles. That's for the eclipse stage. Next is assembly. So assembly is where these new structural proteins and viral genomes would be packed together into new nucleocapsids, as we can see right here in this illustration. And then next is release. So this happens when these new virus particles would exit the host cell, either through direct release, so exit lang basta yung bagong virus particle to the outside of the host cell, or it could happen through apoptosis. Now in apoptosis, as we can see right here, sinira nila yung structure ng host cell to let themselves out. Now if, for example, the virus is enveloped, they could undergo budding. Alright, so here in budding, as they exit the host cell, mangunguha pa sila ng piraso ng membrane ng host cell para yun yung gawin nilang envelope. So, and here are the steps in the viral replication cycle. That ends our discussion for week 11. There were no other references that were used in the making of this presentation other than Mahon 7th edition. This has been Aurel Enriquez. Thank you so much for watching.